All right, in this video I wanted to talk about a process that affects two of the traits we use to differentiate between different phylum of animals when we talk about animal diversity and evolution. So the process we're going to discuss is gastrulation, and we'll see that it affects two different traits, again, that we use to differentiate animals. So first, let's look at the animal phylogenetic tree. We've looked at this in some other videos. And I want to point out the traits we're going to talk about after we understand this process of gastrulation. We're going to talk about the traits of being either triploblastic or diploblastic. Okay, so gastrulation will result in animals that are either triploblastic or diploblastic. Okay, we'll see how. And then we'll talk about animals that are either protostomes or deuterostomes. All right, so again, gastrulation will affect if these animals are protostomes or deuterostomes, and we're going to see how. And that will help us fill in these dots in two more places on our tree. So let's get started by thinking about gastrulation. Again, gastrulation takes place during embryonic development, and it takes place when an animal embryo is nothing more than a ball of cells. So in this cartoon, this ball here represents the blastula. Okay, it's just a sphere of cells. Think of like a beach ball, where the outer layer of the beach ball represents all our cells, and the middle layer is empty, or the center of the beach ball is empty. All right. So the blastula undergoes this process of gastrulation. And gastrulation is when part of the blastula invaginates. So you can see this part of the blastula is pushing inwards, forming a cavity. You could again think about this happening to a beach ball. If I took a beach ball and I grabbed it with my hands, I could take my thumbs and press them inwards to fold the ball over on itself. And that pressing inwards with my thumbs would cause a process very similar to gastrulation. So once this cavity is fully formed and we've pushed this layer of cells all the way in, we form this structure here at the bottom called a gastrula. Right? So it has a layer of cells on the outside, then it has this layer of cells we've pushed into the inside. So let's look at another diagram that represents the blastula and the gastrula. And this one has a bunch more labels we can apply to the results of gastrulation. So again, here is a blastula. It's just a sphere of cells. They're all in one layer and it's empty in the middle. The space in the middle, the empty space, is called the blastocele. Okay, so the space in the middle. Again, when we go through gastrulation, the cells here in the bottom are going to push upwards into the blastula, forming the gastrula. So we've pushed upwards here. We've pushed some of these cells into the inside, while the other cells remain on the outside. The hole we create is called the blastopore. Right? So by pushing in the cells, we create a hole. That's the blastopore. And again, we've created two different layers of cells, the cells on the outside, and now we've got this layer of cells on the inside. We give those cells different names, or those layers of tissue different names. On the outside, we call it the ectoderm. Okay? On the inside, we call it the endoderm. So I see the precursor of a word ecto. Ecto to me means outside, and endo means inside. The derm refers to the tissue. So again, there's ectoderm, the outside tissue, and there's endoderm, the inside tissue. All right? So we've created the outer layer of tissue, the inner layer of tissue, the hole where you can access the inside here, that's the blastopore, and we've still got this blastocele, the space in between the cells. All right. So that's gastrulation. Again, this process is going to lead to those traits of being either diploblastic or triploblastic, and then either being a protostome or a deuterostome. So let's start with those first traits. Diploblastic versus triploblastic. So here's another diagram that represents the difference. If we draw the gastrula, and in this case we've removed the hole, the blastopore. Here we're just drawing the tissue layers. Again, here on the left we've got an endoderm layer, the layer of cells that are on the inside, and an ectoderm layer, the layer of cells on the outside. Some organisms only develop those two layers of tissue. Diploblasts have two layers. Di means two. Diploblasts have these two tissue layers, just the endoderm and the ectoderm. So if we wanted to define a diploblast, it's one of these embryos after gastrulation that has just the endoderm and the ectoderm with no additional tissue in between them. So the space in between is empty, or the two layers of tissue directly contact one another. So how is a triploblast different? Well, tri means three. These organisms are going to have three layers of tissue. So they still have the endoderm on the inside, they still have the ectoderm on the outside, but in between the endoderm and the ectoderm, they have an additional layer of tissue, which we call the mesoderm. Mesoderm just means middle layer of tissue. So triploblasts have a mesoderm layer in between the endoderm and the ectoderm. So if we look at the development of an animal embryo, we look at it after gastrulation, and we can ask this question: how many layers of tissue do they have? Do they have just two, the endoderm and the ectoderm? Well, then they're a diploblast. Or do they have three, the endoderm, the ectoderm, and the mesoderm in between? If so, they are a triploblast. All right, so that's one way we can differentiate animals evolutionarily. Another way is to think about these terms either protostome or deuterostome. Again, these are also related to gastrulation. So I've got another image where we're showing the cells, the embryo before gastrulation. Then here we're showing you the invagination, formation of the blastopore with the endoderm and the ectoderm. This embryo is developing mesoderm here as well. Right. But when we talk about protostomes and deuterostomes, we're really focusing on this blastopore and what happens to it later in the organism's development. This blastopore is one of the holes that accesses the organism's digestive tract. And in protostomes and deuterostomes, these organisms always have a complete digestive tract, meaning there's an entrance and an exit, the mouth and the anus. And again, the blastopore forms one of those holes to access the digestive tract. So it can either form the mouth, or the blastopore can form the anus later in the animal's development. Again, protostome or deuterostome tells us which structure that blastopore hole forms. All right, so in protostomes, that blastopore ends up developing into the mouth, and a second hole develops later to form the anus. Whereas in deuterostomes, that first hole, the blastopore, forms the anus, and a second hole develops later in development to form the mouth. So that's how we differentiate between protostomes, where the blastopore forms the mouth, and deuterostomes, where the blastopore forms the anus. Now those can be a couple difficult vocabulary words to memorize. I had a student in one of my classes several semesters ago who found a way to help himself remember. He simply said, oh, I remember it like this. Deuterostomes form the anus. The anus involves doo-doo. He was just simply connecting a word that he could remember to this more difficult piece of terminology so that he could remember which structure it formed. So Deuterostomes form the anus. Protostomes, the other one, that blastopore forms the mouth. So let's go back to our evolutionary tree of animals and just point out one more time where these traits are involved on the branches. So some animals are diploblastic. They only have the endoderm and the ectoderm, and that is here on this branch. In fact, the only animals that are diploblastic are the cnidarians. The remaining animals up here are all triploblastic. They form that mesoderm layer in between their endoderm and ectoderm. And that's everything from the mollusks up to the chordates. Okay. Within this clade here, where all the organisms are triploblastic, some of them are deuterostomes, while others are protostomes. All right, so remember, deuterostome is where that blastopore opening forms the anus, whereas protostomes are animals where that blastopore opening forms the mouth. So deuterostomes include chordates and echinoderms. We are chordates, humans are chordates. So in our embryological development, that blastopore hole ends up developing into our anus, and our mouth develops separately later on. All right, so that's where we'll end after defining those two more terms that we use to understand animal evolution. As always, I hope you ask questions if you have them, and I will talk to you again soon.